Hey guys, Miss Briggs here. Today we're going to talk about the structure of DNA. So last week we talked about the history of how we found out that DNA was the carrier of genetic material. And so what is this DNA that we are talking about? We do know a lot more about the DNA and its structure at this time. So we'll talk about what the different components of DNA is and how they form the structure of DNA today. So let's get started. So DNA is composed of four types of nucleotides. So what is a nucleotide? A nucleotide, there are three parts to this nucleotide. And a nucleotide is basically like a puzzle piece. So we have these singular puzzle pieces here, and they all kind of fit together. And we'll talk about how they fit together in this video. So one nucleotide is just one of these pieces of the double helix. And so it's going to contain a phosphate group, a sugar, and then a nitrogenous base. And most of what we learn about when we talk about a nucleotide is going to be this nitrogenous base, and we'll talk more about those as we go along. But the phosphate group and the sugars are going to compose these outer parts of the double helix here. And then these nitrogenous bases are going to form the rungs. So there are four bases of DNA, and that's these nitrogenous bases. There are four different kinds. There's cytosine, there's thymine, there's adenine, and there's guanine. And you can see up here we have like different puzzle pieces that fit together. So these bases are going to fit together in certain ways. So before we get into that, let's talk about Chargaff's rule. And this is the amount, basically the amount of thymine equals approximately the same amount of adenine, and the amount of cytosine equals the amount of guanine. So when I think of these, it helps me remember, it might not help you remember this, but I think of a G and a C look similar. They're going to go together. And then the thymine and the adenine are going to go together. So you can see that they kind of fit together like these puzzle pieces, and we'll talk about how their bonds form, how they bond together as well. But just think about these rungs as only the guanine is going to go with the cytosine, and only the adenine is going to go with the thymine, and vice versa. Thymine is going to go with adenine, cytosine will go with guanine. You'll never see cytosine with adenine or guanine with thymine. So this is called the base pairing rule. So when nucleotides pair, they always pair with their base pairs. So right here we have a molecule of DNA, and we have the sugar and phosphates are making this backbone here. And then our nitrogenous bases are making these rungs, and so they're connecting on these rungs. And they have different connections or different bonds between the different base pairs. So you can see here in this diagram that cytosine is going to form with guanine, adenine with thymine, and so on. So these base pairs are joined together by a hydrogen bond that connects the bases in the center of the double helix. So you can see a little bit already how these different base pairs form different hydrogen bonds. So they're going to be attracted to each other, and the cytosine and the guanine are going to form these three hydrogen bonds, whereas the thymine and adenine are going to only form two hydrogen bonds. So they're bonding differently with each other, which is why the adenine and guanine couldn't bond together, and the thymine and adenine do bond together. So let's take a little bit of time to answer some critical thinking questions here. So if we had a base sequence of thymine, guanine, adenine, cytosine, thymine, adenine, what would be their pairs? So go ahead and stop the video. Think about what would pair with these bases and write that in your notes.